Hello everyone, my name is Thalissa, and today I will be presenting some topics in quantitative genetics. Professor Augusto Garcia presented the proposal for this video in one of his classes in the graduate program in Genetics and Plant Breeding at the Luiz Queiroz College of Agriculture. You can access the content of these videos through the link in the bio. I will be covering the genetics background, and Diana and Angelica will drive you through the decomposition of the genotypic value. So, let's start our class. To understand quantitative genetics, we must briefly review some things we already know about genetics. Let's start with Gregor Mendel, the father of genetics. The phenotypes Mendel observed in peas were all or none. A pea seed was either round or wrinkled, yellow or green. But Mendel had no idea about everything we constructed based on his work. However, our main interest in plant breeding is quantitative inheritance traits and how they are passed on to the next generation. But how the quantitative traits are distributed? Earth length in corn, for example, it is a polygenic phenotypic trait, controlled for more than one gene, and shows a continuous variation. The quantity or biological values distribution around the mean follows continuously and precisely. So imagine the characteristic related to plant disease, photosynthesis, size, age, weight, or drought tolerance. They are all controlled by many genes. But what happens if a plant breeder wants to increase the yield? He probably will have to improve the plant simultaneously, which is quite challenging. They have so many genes and infinite distributions that they are not too easy to work. Therefore, it is necessary to understand the allelic and genotypic frequencies. Let's consider in our example a population of colored bean seeds, which have different phenotypic classes and are controlled by a gene with two alleles showing complete dominance. Suppose we have 50 red beans, 500 pink beans, and 450 yellow beans, resulting in 1,000 beans. For the calculation of genotypic and allelic frequencies, we must remember what we call the Hardy-Weinberg equilibrium, which says that in a population that has not undergone migration, mutation, or selection, both allele and genotype frequencies remain constant through generations. In addition, the population will be in equilibrium if it does not suffer genetic drift, so the matings were random. Therefore, to calculate the equilibrium, we need to add the p-square, which would be the homozygote B1b1, multiply the value of the heterozygote B1b2 twice, and raise the value of the homozygote B2b2 to the square. The total of the sum should be equal to 1. Understanding this, we can move forward for the next steps related to the locus influence on the phenotype.
We must remember a very important relation in quantitative genetics, which explains that the phenotype is a result of the genotype plus the environment effect. In this video, we will be focusing only on the genotype and how it can influence the phenotype of an individual. Fisher proposed a very simple example to understand the effect of the genotypic component. Let's imagine a diploid individual that contains only two alleles in a single locus that express a certain trait, B1 and B2. The possibilities of genotype formation are the B1-B1 homozygote, the B1-B2 heterozygote, and the B2-B2 homozygote. These three genotypes can be represented on a linear scale with the metrics that we brought here. On this scale, 2a represents the difference between the homozygotes B2-B2 and B1-B1. That's it, the scale size is 2a. We have the value of 0 at the beginning of the scale and in the middle the value of a. Fisher then tells us that the B1-B2 heterozygote will not necessarily occupy the middle of the line. For Fisher, the position of the heterozygote is a function of A, which denotes additive effect, and the value of K, which denotes the value of dominance. Within this context, we can see that when K is equal to 0, the heterozygote will be occupying exactly the middle of the line, so that B1 and B2 behave in a completely additive way, since there will be a lack of dominance. If the value of K is between 0 and 1, the heterozygote will not longer occupy the middle of the line. Due to the partial dominance effect of the B2 allele will have over B1, bringing the heterozygote closer to the B2-B2 homozygote. If the K value is equal to 1, the heterozygote will express the same effect as B2-B2 homozygote, due to the effect of full dominance of the B2 allele over the B1. Still, it may happen that K has a value greater than 1, which explains the effect of overdominance of the effect of B2 allele over B1. The opposite is also true. If the value of K is equal to -1, it means there is a full dominance of B1 allele over B2, as well as values lower than -1 explains the overdominance of the B1 allele over the B2. The authors of the 1998 book named Genetic and Analysis of Quantitative Traits, Lynch and Walsh, emphasized that the definition of 0 for the B1-B1 homozygote was only performed to facilitate the understanding of the additive and dominance effect of the alleles. We need to be aware that the 0 does not indicate the gene in question does not add anything to the total value of the trait. This is because the genotype value is still influenced by the environment effect, and the intercept in this scale is the parameter that will tell us from which value it will start.
Let's start talking about one of the Fisher's most important advances in his paper published in 1980, the partitioning of genotypic variables. In several components, remembered the previous concept, the graph can be divided into dimensions, where Fisher proposed a linear regression model that calculates the number of copies of the favored allele, as indicated on the x-axis, with values 0, 1, and 2 copies for a diploid. In y-axis, I represent the genotypic values for the homozygous, has a scale value between 0 for genotype B1, B1, and twice A for genotype B2, B2. Now, the heterozygote, where will be located, as we discussed previously, its location will be defined by the equation 1 plus k basis A, which indicates that this value will depend on the activity and dominance exhibited in the genotype. The solid points represent the genotypes B1, B1, B2, B1, B2, B2, B2. If we look at the distribution of the points, this represents the true genotype values for each individual. Now, we plot a line as a function of homozygous and heterozygous individuals to study the factors that may influence the response variable. As a first approximation, we will fit a linear model with a least square regression between the genotype values in response to the locus B alleles. Looking at the points on the line, we can determine that these are the expected values from the model. Now, if we focus our attention on the heterozygous, we can observe that the expected value allows us to quantify the deviation with respect to additivity, and the difference between the expected and the observed values represent the dominance deviation. Now, making use of the previous concept, we can study with more details the multiple regression model proposed by Fisher. We can observe that the genotype value Ig is defined by the estimated genotype value plus the error. If we expand the formula, we find that G hat can be defined as the population genotype mean, alpha 1 by the number of copies of allele B1, the sum of the effects of each allele in the genotype. Simplify the formula with the premise that we are working with a diploid. We can infer that N1 for N2. Proceed to replace in the formula the below the product, and next step we can define I as an intercept, which will be equal to the population mean plus twice the slope 1. Now we can determine that the regression coefficient alpha is equal to alpha 2 minus alpha 1. Finally, we have an univariate model related with the Jij value. With this in mind, we can see that from a multivariate model, we can obtain an univariate model depending on the estimated genotype. For example, in the case of heterozygotes, this is going to be equal to the population genetic means plus the sum of the substitution effect of each allele in each different genotype class. Now, the question will be how to calculate alpha 1 to alpha 2. With the help of some properties of the expectancy of the value, we can determine that alpha 2 depends on the B1 alleles and that alpha 1 depends on the B2 alleles. In summary, this effect depends on the effect of allelic substitution. Now we have a new parameter to calculate. Then, the allelic substitution effect is calculated for the regression line slope coefficient, which is equal to the covariance between the response variable and the dependent variable, divided by the independent variable's variance. Again, using some of the properties for expectation value, we have that the effect of allelic substitution will depend on the net effect due to additivity. K determines effect and the frequency of allele B1 and B2 in the population.
By convention, we will assume that the B2 alleles contribute positively to the expression of the trait of interest, while B1 is unfavorable. This graph shows the influence of the allele frequency of B2 and the dominance effect denoted by K on the genotypic value. On the x-axis, we have the number of allele copies, while on the y-axis, we have the genotypic value as exemplified earlier in this video. The slope of the regression lines capture the average additive effect of the two alleles, which means that it shows the part of the genotype that will be inherited by the next generation. Let's observe in the graph how the regression lines behave as we change the value of dominance and allele frequency of B2. When the K value is equal to 0, it means there is no dominance effect. Thus, we can observe that the slope of the line does not change as we change the allele frequency. Furthermore, it is important to note that the line passes exactly over the points, which means that the predicted genotypic value is the same as the observed ones, and the line capturing the entire additive effect of the gene. Now, let's set the allele frequency to 0.5 and just change the dominance value. We can see that the slope of the line is not changing, but the deviations are increased as the dominance value increases. The greater the deviation, the greater the bias of the quantitative estimation of this trait, and the lower the predictability of the model. We can also observe that in this situation, only the intercept changes. This is because the statistical model assumes that the slope, denoted by alpha, is a function of k multiplied by the difference in allele frequencies. As the frequencies are the same, the k is null, leaving only the value of the intercept. Another important thing to remember is that dominance occurs from the interaction of the two alleles, and that it is not inheritable by the next generation. That's because the individual in this example is diploid and sends only one allelic copy to the next generation. Now, when we set the dominance value to 1 and keep the frequency at 0.5, we have the situation that the B2 allele has full dominance over the B1 allele, causing the heterozygote to behave the same way as the homozygote. By decreasing the frequency of B2, we observe an increase in the slope of the line. This is because in this situation there is a high frequency of unfavorable genes, which do not contribute to expression of the trait of interest. The increase in the slope of the line shows us the effect of the allele substitution of B1 by B2. In terms of genetic improvement, from the first crossing cycle, significant genetic gain is expected due to the effect of replacing unfavorable alleles with favorable alleles, which contributes to improving the trait of interest. On the other hand, when we increase the frequency of B2, we notice that the slope of the line decreases. This is due to the low effect of substitution unfavorable alleles with favorable ones, since the population already contains a high allele frequency of favorable genes. If we use the value of k equal to -1 and increase the allele frequency of B2, the complete dominance will be of the B1 allele over the B2. Thus, we observe that the genotypic value of the homozygote B1 and the heterozygote is 0, as the dominance of B1 only allows the additive effect to be observed in its absence.
Part of the supplementary material of this discipline, and it will allow us to understand in a dynamic way the variance decomposition proposed by Fisher. In this case, we are going to see how the total genetic variance and its components, additive genetic variance and dominance, interact depending on the k values that represent the type of dominance. Now, let's start in the simplest case. In the absence of dominance, we can observe that the additive and dominance variance lines overlap, indicating that they are equal. The second case we are going to explore when k equal to 1. Then we can observe that there is a decrease in the additive variance and increase in the dominance variance. This is due to the fact that the total variance is the sum of these two factors. It is important to note in this case that when the frequency of alleles B2 increases considerably in the population, the dominance variance sets the additive variance. We also observe that the total genetic variance decreases significantly in the population. To understand the concept allele beta, let us approach a practical case in which a population has been subjected to a process of exhaustive selection. For a character in this situation, the genotype variance is considerably decreased, and the percent of genetic gain in each generation became small and small. Let us remember that the dominance effect can take negative values. In this situation, we will have that the B2 allele will have dominance over B2, but the concept will be the same as the previous mention.